Good afternoon. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the beautiful inland Pacific Northwest. Today is the 3rd of November and the year is 2021. We are going to do the probably second to the last lecture on aging and the comorbidities that lead to mortality in humans. So I say possibly this is the penultimate because there might be at least two more, but I think maybe only one more depending on how far we get. So I hope that we can get to where we're essentially ready with only one more after this. So that's where we're going to try to run to, and hopefully we'll get there. Okay. All right. So what am I calling this particular slide presentation? I'm calling it the Dialectical Event Ontology. I know that's something I've said many times, of immunosenescence as a harbinger to aging and the sickness unto death. Of course, those are associations with Soren Kierkegaard, that last part of that statement. And here in Kierkegaard's book, which I recommend, he's talking about despair as the sickness unto death. Here I'm being a little bit more physiological and generic and not so much talking about something neuropsychiatric, although we will get into some of those discussions in aging inevitably because we'll talk about neurodegeneration, right? So again, I'm Dr. Dan Guerra, and this is Authentic Biochemistry. And I'm going to call this the CODA because this is going to be the ending, as I said, of our long arc of discussions, now a year long, into human aging. And what results in the aging process ultimately is a series of comorbidities, which causes illness and sometimes frank disease, and then ultimately, of course, the death of the human. So I wanted to get into the pathobiochemistry, pathophysiology for a long time on this subject, and that's what we've been doing. And finally, I'm going to do the synopsis. All right, enough prolegomena. Now we can just get right into it, right? All right. Now, I want to remind everyone what I mean about this dialectical event ontology, something that I've put together as a new way, a new paradigm in understanding biological systems, particularly the kind that I study, which are humans and the aspects of biochemistry in humans as it relates to their physiology and also disease state. So let's take a look at this dialectical event ontological method, which I've generated. I'm calling it an adaptational, temporally processive investigation of biochemical and physiological events, employing a dialectical method to expose and hopefully therefore interrogate oppositional, empirical, and rational evidence through the scientific method that includes hypothetical deduction and inductive synthesis towards an event ontological knowledge architectonic. A little bit more explanation. There is a well-established immunoepigenomic tailoring of neural, endocrine, digestive, cardiovascular, adipose, muscle, and metabolic responses to the micro and macro environment through classical constitutive surveillance and acquired effector, cellular, and humoral defense mechanisms using reversible covalent modification and hydrophobic interactions of the basic organic compounds we find in the cell: nucleic acids, carbohydrates, proteins, and most demonstratively, lipids. So one more aspect of this paradigm in this narrative presentation of it. The dialectical event ontological method, then, is a synthesis of these phenomena that I've just mentioned toward a central molecular theory for free will driven, agency based, existing individual adaptation and assimilation that hopefully will better explain the core event ontology of human development, decline, and death at the molecular level. And we hope to obtain with all of this discussion, we hope to obtain a sharper grasp, or we hope to apprehend physiological processes that become chronically compromised during aging. Okay, so that's kind of a long-winded series of definitions about what I mean by my method, and that's the method gets employed throughout all of my lectures in authentic biochemistry for the last several years now that I've been doing this. Now, what else I want to say about this? Using a method that centers on stark detail of heterotrophic biochemical phenomena. By heterotrophic, I mean those of us, animals, that have to consume preformed so-called essential nutrients from nature so that we are not autotrophic. We don't synthesize de novo from inorganic structures, organic compounds, the complex structures that something like bacteria and plants can do. Heterotrophic is involved in the agency aspect, and the agency aspect of human endeavor also must include this discussion of free will. If I don't include that in my series of definitions in the way that I develop this theory of dialectical event ontology, I would then be absenting a huge area of understanding of how each individual moves through life, dealing with the vagaries of the environment, the composition of the genome, and the interactive complex of the immuno and neuroimmunological axis that ultimately derives the self, the human, through the living and then through the aging. And the longer the aging occurs, the higher the morbidity increases. Ultimately, these morbidities will advance to such a stage that they will become mortal and death will obtain. Okay. So that's a long thing talking about heterotrophic. So heterotrophic biochemical phenomena within cellular networks controlled by individual agency within a changing environment requires, in my opinion, the analysis of the spatiotemporal dimensions of free energy flux. So there would be a discussion of enthalpy and entropy, for example. And what obtains from this approach is a living event ontology. Human aging, in my estimation, is the unique paradigm here. It's unique because of the aspect of free will. Now, I keep on emphasizing free will, although that is a term from theology and philosophy. It's also a term in psychiatry, psychology, and indeed in neuroscience. So, whatever side of the aisle you are on in terms of whether or not free will exists, I'm going by the standard that it does indeed exist. And my evidence for its existence relates to my study of biochemistry in living systems. Each individual making selections, moving through life with judgments, decides how they interact with their environment. That interaction ultimately will generate an epigenomic pattern that will result in that individual's exposure to and surveillance and perhaps elimination of potentially toxic substances and dangerous pathogenic organisms. Even after successful removal of any of these pathologies through the lifespan, because of the immune system, both the innate and the acquired functioning together, generating memory cells, ultimately in the T and B cell lymphocytic lineages, there will be left both an indelible and a plastic response that will resurface upon re-challenge of a given specific organic chemical compound signature or perhaps a pathogenic molecular signature. This is what I mean by agency being important, because the agency runs through from the beginning of life all the way to the end, and that agency gives you the unique status of interacting with the natural environment in that very specific way, thus generating the living system that ultimately has these morbidities and then, of course, leading to death. So with this kind of paradigm, I'm arguing for an individual basis for understanding these morbidities and mortalities. Now, of course, I'm a scientist, so I understand that we can do an induction of all of this collected individual data. And that's what we do in authentic biochemistry. We make inductions. In fact, the whole theory of looking at event ontologies and using a dialectical approach or method stems from my interpretation and understanding, and ultimately my exposition of the published research in my discipline of biochemistry, and then related disciplines like physiology, immunology. So this is where we're coming from. This is where we're going to end up, both in these lectures and, from my suggestion, where we all end up in the end. Anyway, so let's talk about the diaventome. So for that, I'm going to put on my glasses. Notice that it's a trigonal planar association. And what do I say about this diaventome? I say that there are biological networks and they are processes. They become eventual in nature. So they're not substances. So as opposed to a substantial network, we're talking about eventual networks, processes that occur through time. This gives me an event ontology that reaches across this biological network of genetic and environmental interaction. And it is articulated and synthesized through epigenetic modifications of the classical and acquired immune system, thus creating a responsive plastic and indeed elastic memory field capable of metabolic scanning, focusing, accession, mediation, learning, and reacting to stimuli through time. Now, again, I want to remind you what I mean by plastic and elastic. When we have a plastic response in living systems, it means that there is an induction or there is some stimulus, and the impression left from that stimulus changes the molecular architecture in some way, maybe diverts a metabolic pathway, for example, and leaves that impression for some time after the event. Okay. Whereas an elastic event leading to perhaps a shorter term memory field in the living system is one that is read into the system. Then, after it's read into it, it can be erased. So it's elastic. That means it is easily reverted back to the native state. Now, I say that with a huge caveat, because even with an elastic response, you still leave some molecular signature behind. That's because all of the host of modifications that have occurred in the living system will leave some nascent, ultimately persistent molecular memory because of pattern recognition that occurs in receptors, for example, throughout the central and peripheral nervous systems, as associated with the innate immune system, as well as all the signal transduction cascades we see in biochemical systems. Now, a word about the environment. The environment, okay, so you have the genome. This is what you inherit. Then you have the environment, which is anything but neutral, anything but static. The environment is dynamic, right? So what I say is the existing individual agentically interacting through the events obtained across the inherited genome, the changing environment, and the receptive architectonic neuroimmunoepigenome. This has to occur through time, therefore it's not a substance or a series of generating concentrations of known metabolites and looking at the relative molarity of those substances in given cells, for example, or in circulation. But the event of the changes and alterations of those millions of different molecular signatures happening in millisecond to second time sequences. That's the kind of event ontology that is leaving the overall impression on the body, leading to and associated with the aging process. So ultimately, I want to say this. Biological phenomena involve the a priori synthesis of a reception and signal transduction cascade network. You can, if you like acronyms, we can call that the RSTCN. Now, again, a word about these definitions. A priori synthesis means before it occurs. How can you get a synthesis before something is transmitted? Well, by reception. You have receptors that are part of the genome, which are, in the end, lipoproteins or proteins or glycoproteins, that is, bare proteins, lipoproteins, glycoproteins, for example. And those receptors respond to the stimulus, right? So an a priori synthesis is how the body reacts to the wave particle stimuli that occur during the living process. And that comes from the environment interacting with the genome through the processing of the immunological circuitry, which is always engaged throughout life, and then the epigenetic alterations of those signatures, which can become epigenomic, meaning they can be stably inherited, at least through cell division. At least through cell division. Yeah. Okay. So biological phenomenon involve the a priori synthesis of a reception and signal transduction cascade network. This pattern of cohering events appear as energetic and mass dynamics much like chemical bonds. So the dialectical interactions between environment and inheritance, genome and environment, event a trigonal planar axis, which is, I think, the simplest way of understanding it, a 3-point axis. Ontology via stochastic, means pseudo-random, epigenetic retailoring of a biological communication system using as its method of communication the immunological domain. Okay. So I'm not going to apologize for giving you all this as prolegomena. I'm trying to do a synthesis of the aging process. And for me to do that, I want to introduce my whole way of understanding what I've learned in this year-long trek through the literature of pathobiochemistry and pathophysiology and disease in aging humans. Okay. All right. So, because I feel like when I talk about this, I'm preaching to them, just lecturing. Okay. So, what are some basic dialectical events in biochemistry and physiology? Just basic, when I'm talking about dialectical, meaning we can describe when they're on or off simply. There's actually a more complex way of looking at it. I call that either or. And we will develop that as I go on. So, molecular and cellular events that occur dialectically across mechanisms that result in real-time modifications in changing to two changing conditions. So the mechanisms include, but they're not limited to, replication, recombination, and repair of DNA. I call that the triple R, right? That's what I used to call it in graduate biochemistry lecture. Also, number 2, transcription, alternative splicing, and then translation of RNA, obviously to polypeptide. Signal transduction, third event. Membrane fluid dynamics, which includes both biochemistry and biophysical alterations. Cell fate repertoire, which include autophagy, apoptosis, senescence, and necrosis. There are multiple subtypes of at least apoptosis and even senescence, but we will develop the florid nature of those as we go on. Also, another important dialectical event, developmental programs, including embryogenesis, and then the cellular division and differentiation, including adipogenesis, neurogenesis, myelocytogenesis, lymphocytogenesis. These are just a few, right? Gametogenesis for inheritance, right? For sexual reproduction. And of course, oncogenesis, a really important thing in the aging human. As I say, etc. Okay. I didn't want to make that an exhaustive set of terms right here. So here's a dialectic. Here's how I proceed with this method. Okay, that's what we're going to do here. T lymphocytes react in the intact human dynamic event ontology to respond to the environment, maintain cellular and physiological health, and to prepare for future change that includes such heterotrophic interactions as nutrition, neurological imprint. So nutrition would be diet, neurological imprinting, that would be like generating new knowledge bases. Hopefully what you do with authentic biochemistry, for example, or when you go to any good lecture. Disease could be caused by abiotic or biotic pathogenic incursions. And of course, aging. Aging is itself a component of this dialectic. but it is impacted and it is generated by the complete trigonal planar understanding of genome environment and the immunoepigenome, okay, including the central nervous system and the peripheral nervous system. So there's a neurological component. I keep wanting to emphasize that. Now, back to T lymphocyte. T lymphocyte lineages and associated biochemical communication are modified, as we've been talking about for at least the last 6 or 7 lectures in audio podcast, are modified via changes in the epigenome. That means methylation, acetylation, ubiquitinylation, those covalent modifications we've talked about for either cohering DNA or cohering histones, as well as canonical, inducible, and also repressible gene expression and membrane recombination. Key epigenetic molecular events in this dynamic process include chromatin remodeling, I've been now calling it retailoring, CpG methylation, histone acetylation, and of course microRNA interference, which require the establishment of a event ontological phenomena. So those molecular interactions establish the phenomena that I call a dialectical event. And because it's in real time in living systems occurring, I call it an ontological event. Right? Okay. So each of the words I'm using, I'm not adding them together to add hyperbole. I'm trying to actually be as discrete as possible in explaining my terms. Okay. All right. So let's look at this paper in Frontiers of Aging Neuroscience 2020. Why not? Age-associated neurodegenerative diseases like Alzheimer's and Parkinson's disease have become significant endpoint in the national health. Cognitive decline obviously can occur at any age. We've seen it. We've all probably experienced it at some level. And this happens at the phenomenological level. So it's a readout. It is a presentation. But intellectual deficits are not equally distributed. So it is incumbent on the research community to differentiate the age-related neuropsychiatric decline from other forms of CNS disease. Okay? Because there are different nuclei in the brain that are more associated with intellectual endeavor than, for example, motor skills. Driving a car, playing a piano are different than trying to solve mathematical theorems or to understand how 8 different biochemical pathways interact coherently in a cell. Okay? So, age, or we can call temporosequential dementia, obtains prodromal. So we've seen this in the literature. We talk about Alzheimer's disease in particular, and we say, what are the early signs of AD? What are the early signs of Parkinson's disease? Right? And so there are actually psychiatric tests for this. How well you can remember a series of numbers, how well you can fit in a number in a series, how well you can count backwards, how well you can explain what you've read in a dictionary definition in your own terms, things like that. Cognition, general cognition. Okay. So those prodromal issues are very important because underlying those presentations, which are basically intellectual failings when you can't perform the task that was originally set up for you to perform, such as inserting a number in a series and explain the sequence of numbers you're given. The underlying all of that is biochemistry, biochemical pathways, and cellular metabolism is altered prodromally long before you see neurodegeneration, frank, in certain nuclei in the central nervous system. For example, sometimes before age 70, you get this prodromal dementia. And that can happen in otherwise non-compromised adults. However, vascular diseases and obesity, now I'm bringing in more of what we know to be contributing to not just neuropsychiatric disorders such as neurodegenerative complex, but also all of the vagaries of the morbidity and then the illnesses that are generated in people when they are younger during those prodromal phases. And when I'm talking about prodromal, I don't just mean neurodegenerative diseases. I mean any disease, including that major chronic disease we call aging. Okay. A lot of that can be associated with earlier stage vascular diseases, such as atherosclerosis, and obesity as an overarching disease state, which can lead to chronic inflammation and a complete corruption of bioenergetics. And because of that, immune response to potential pathogens and xenobiotics. And of course, the whole nutritional aspect of the loss of sensitivity to adipokines, generating the awareness of being well fed or the appetitive nature of continuing to eat long after the stomach is filled. All of that is generated via a neuroendocrine system, right? And all of that starts to fail upon the complexities of obesity. So, proteomics and lipidomics, now we're talking about method here, have been implemented to identify serum biomarkers, prodromally used, for this early diagnosis of dementia. And unfortunately, I would argue that they have met with withering success. Why that is could be an axis of two things. One is we don't have enough information yet. And if we build a large enough library, we might be able to triangulate and get a better focus on biomarkers as they change as events through time rather than as substance found in the blood. When your blood is drawn, you do a panel once or twice in a year or once or twice in 10 years. And depending on what that panel says, your physician, your primary caregiver say, oh, there's nothing wrong there, you look fine. But if you had that same panel done multiple times, you'd be picking up not substances but events, you see. So there's one reason why I say there's withering success. The other is we don't really know what to look for. So we're looking at standard, often serum biomarkers or whole blood biomarkers, because most often we do not want to do biopsies, certainly not biopsies that go into the central nervous system. Even things like spinal taps are not something someone's going to volunteer and say, give me a spinal tap. I want to know if I'm prodromally going to develop early stages of Parkinson's disease. Some people may opt for that or, get in the front of the line to have that done, but most people won't, right? So those are two reasons, I think. One is we don't have enough information about what we already have accumulated, knowing how things interact. And the other is we may never basically know because we don't really know what we're looking for. We need to be looking at events, not substances. Much more difficult to measure. Okay. Now, the key to aging morbidity and aging morbidity are not synonymous, although some may argue the older you get, the more moribund you get. We know, because we live in the environment and we're agents and we're out there in the world, that there are different stages of aging process for different people. And that process is not absolutely temporal. There are alterations in that process. And we yet to understand that. We would argue there's a genetic predisposition. We might argue that, well, if you live in a very unhealthy environment with a lot of pathogens, or that is unclean water, unclean sanitation. But even when we look at that system, we can agree that people that live in very squalid environments, nevertheless, can live well into their 70s, 80s, and 90s. And people that live in really clean environments and have regular health care can develop diseases and become very ill in their 20s and 30s. And one might argue, well, it's life habits, life choices. I would agree that's part of the free will component, and that it has to be added in, right? And that's why I did add it in. But also we have to understand that it's the interaction that matters, the interaction with the genome, whether or not there's an epigenetic change, and whether or not the immune system has been disturbed, leading to either a plastic or an elastic response. Right? Okay. So, see, the reason I gave you all those audio lectures these last several weeks, and actually for the last year, and now all of these is video, is because now I can talk much more efficiently about these systems because I've given you much more florid detail in all those previous lectures. Right? That's why I did that. So now I can call upon your memory for you to be able to follow along with what I'm saying, and so that there's actually communication between myself and the wonderful people that are interested in what I produce. All right. So here's detail. Now I've shown this slide many times in lectures. So to most of you, I'm sure it's not a surprise to see it again because I have a very small group of people that actually watch these video lectures because I can see the numbers. Now, it's talking very specifically about T cells. Okay, we gave you all this general information, but then I started going into the example of T lymphocytic interactions in aging. So now we're going to go to the next level and talk about what kinds of T cells do we have, very generically. Okay, now this is a movable feast. We're learning more and more about T cells all the time. They tend to be the magnus opus, in my humble estimation, of the immune system. Not the B cells, the T cells. Okay, and I'll explain why, and I've already explained it several times, but I'll go through it again and at least generate my argument why I think that. That's all I can do, right? Okay, now we can talk a lot more about this, but we're going to start off with just talking about CD8 positive T cells, CD4 positive T cells. CD8 positive T cells is an expression of a specific protein on the surface of those cells, right? In the presence of the interleukin cytokine known as interleukin 12, will generate increasing T-bet levels. T-bet is a transcription factor that has to be induced upon expression. It's a nuclear gene, right? So CD8 positive T cells in the presence of interleukin 12 will generate a T-bet now expressing cell. That cell, if it has another transcription factor called BLIMP1, now there's many more, but those are two signature transcription factors, will become a cytotoxic T cell, cytotoxic T lymphocyte. So right away you understand that T-bet activity as a transcription factor is context in interleukin 12 and cell type dependent, because if you look at CD4 positive T cells, which are a different subset lineage of T lymphocytes expressing CD4, not CD8, so they're single positive cells, we call them. Again, in the presence of interleukin 12, that cytokine, you will have T-bet expressed, but you will not have this BLIMP1 expressed. Now, there's a lot more that is changed, but those are just two easy ones to see here. Now, A cytotoxic T cell will, in the presence of interferon gamma, aid in the Th1 response of inflammation. Th1 is a different lineage. Okay, it's a helper cell. Cytotoxic T cell is not a helper cell. You get the Th1 cell, which is only one of many helper cells or effector cells from the CD4 single positive lineage. In the presence of interleukin 12, you'll get Th1 when you just have the T-bet expressing. You see? Okay. Now, that T-bet expressing cell without the expression of the co-transcription factor BLIMP1 in the presence of interferon gamma, that is again a pro-inflammatory cytokine, will give you Th1 inflammatory response. And that inflammatory response will also include certain chemokines, CXCL9, 10, and then the receptor CXCR3 and its ligands. So you have chemokine receptors and chemokine, these are again are glycoproteins generated from multiple cellular lineages in the body, including endothelial, epithelial, as well as immune cells. Okay. Now, notice that if you have a CD4 cell in the inducing environment of interleukin 12 and interleukin 6, those two cytokines, you can generate a T-bat expressing cell, which also has this protein BCL6. This will actually be a T follicular helper cell. So now you're starting to see more sub-differentiation of even the T helper cells. So what's a T follicular helper cell? These are very important. For one reason that I can think about just now, just off the top of my head, is you find these crossing the blood-brain barrier. More so earlier on in the aging human than frank T helper cells. T follicular cells can move through the blood-brain barrier at a higher rate because this is where we find them. And we find them also in the cerebrospinal fluid. Spinal tap, right? So T follicular helper cells, or TFHs, are a specialized subset of CD4 positive T cells. They were first identified in the human tonsil. Okay? That's a lymphatic organ, you know. And what these TFHs do is play a critical role in protecting or protective immunity, helping B cells, that's right, B cells, the ones that produce antibody when they become plasma cells, professional plasma cells, helping B cells produce an antibody against foreign pathogens. So TFHs are located in secondary lymphoid organs like the tonsil, including spleen and in various peripheral regions where there are lymph nodes. Okay, so T follicular cells, very important. We find them sometimes in the central nervous system, even in healthy adults. Earlier on, than we find other T lymphocytes, for example. Now, those also will participate in Th1 inflammation. So you have cytotoxic T cells, you have Th1 cells, and you have T follicular helper cells, which are these here. Yeah. Now, those cells can further differentiate in the presence of interleukin 21 and interferon gamma. And what they will do in the process is take this germinal center B cell and convert it to a BCL6 cell. You see? So this has BCL6, this follicular cell. So does the B cell. And it's also going to have an IgG2A generated. Okay? It's your first series of immunoglobulins, right, on the surface. So that cell could be converted to a plasma cell, which is a frank antibody-producing cell. And this is when the IgG2A is secreted. That's what's shown here. Now, this B cell being converted to a plasma cell, which is an antibody-producing cell in the body throughout the periphery, after antigen presentation, antigen attack, and using the recombination of essentially the B cell receptor, right? The VDJ regions. They're going to generate all the different types of IgG, starting with IgM, right? The membrane-associated immunoglobulin. They're going to be expressing two transcription factors, this BLIMP1 again, and the T-BET. So they're kind of similar to cytotoxic T cell, but doing something totally different. They're secreting immunoglobulin 2A. Now, that's really important and significant and rational to this whole discussion, because that immunoglobulin 2A is going to be an important growth factor for a lot of processes. Okay. Now, let's go on. A Th17 cell, and we can talk about where they came from later, it's going to have the transcription factor ROR gamma T. Remember that ROR has to do with retinoic acid orphan receptor. Retinoic acid from vitamin A, that's the ligand for that receptor. That's a transcription factor. When the ligand binds to it and moves through cytoplasm to the nucleus, generating an expression of genes where there is a receptor for that whole system. That's the ROR gamma T transcription factor lineage. Again, in the presence of interleukin 12, it comes up again and again and again, as you can see. We'll give you a ROR gamma T, now T-BET expressing cell lineage. And in the presence of interleukin 17, which is made by this so-called Th17 cell, that's why it was named that, okay? It's called that because it makes a new a new cytokine or interleukin, and that's IL-17, glycoprotein secreted by the cell, specific one, pro-inflammatory typically. And in the presence of interferon gamma, will also aid in the Th1 inflammatory response. This is a full-blown inflammatory response, but it can also be a chronic inflammation from multiple sources, as you can see here, right? Finally, let's talk about Treg cells. Treg cells, Th17 and Tregs and Th1s and Th2s, all those are going to be coming from CD4 lineages, just so you know. FOXP3 expressing T lymphocyte, CD4 positive as well, is going to be expressing FOXP3. FOXP3 is the canonical molecular signature transcription factor for T regulatory cells. Now, why is that significant? Well, you're going to see in a few minutes why. But once you start making T-BET along with FOXP3, these become professional regulatory T cells. And by regulatory, we mean that they inhibit the inflammatory response when they're in their fully active, not in their modulating mode, but in their fully active T regulatory mode. Tregs will suppress the immune response. And that means there's a built-in system-associated cellular differentiating pattern that's going to give you a T cell population dynamic, which will suppress all the other inflammatory responses. So it's a built-in retrograde pulling down, shutting down an inflammatory response. This is absolutely critical, because if the inflammatory response is not erstwhile controlled, once it is activated and stimulated, showing these all these cells up here, all these regional cells here, you'll get a massive pro-inflammatory response. And if it's not checked by T regulatory cells in the location, vicinity of where this inflammatory response is occurring, then you get uncontrolled inflammation, which is going to cause massive tissue death, because that's what an inflammation will cause. It can cause apoptosis. It can cause necrosis. Even some of the subtle chronic inflammatory responses can generate senescence. Okay? We talked about this many, many times in many of the lectures over the last year. The senescence-associated secretory phenotype is one of those patterns. Okay, so now you get the whole picture there. All right. Now we went through this before, but I'm giving you this now complete detail because I'm explaining to you, now we visually are going through this, how the aging process must carry forward all of these biochemical, physiological, cellular event ontologies in order to remain healthy. And if there is an alteration in any of these event ontologies, and already we're looking at many, just right here, you start to pick up a lack of regulatory control, and that can lead to morbidities. And those morbidities, when they become frank, and they are indeed a plastic response where they're left behind after an induction of chronic inflammation, for example, because of obesity linked diseases like metabolic syndrome or diabetes type 2. Those kind of chronic inflammatory responses, then, not dealing with any new pathogens or abiotic organic compounds inducing an immune response, but rather caused now by autoimmune signatures because of alterations in the regulation, because of the chronic condition associated with the aging and choices made by the aging individual, hence the agency of that individual and their free will. You see now, this is how you get very specific phenotypes for disease presentation. And this again explains why there's a withering response to looking for biomarkers. This is all happening within cells. Some of the cells secrete some of these, for example, cytokines and chemokines, and you get that. People look at that when they do blood panels. But is that explaining this whole story? Just a few cytokines, chemokines, growth factors? No, it's not. We need to know what's happening inside these cells, and not just once, a snapshot, right? You had the blood panel draw last May and you're fine, but what's happening over time, right? And not just, every 3 months, but what's happening all the time over time, all the time over time. Obviously, we're not going to measure that, and I have no place to say I would want those things measured. What I'm saying is that that's all the occult, all what we don't know, everything that's opaque about the living system, because it's dynamic, not substance ontology. Yeah, keep on bringing that home, but that's a major emphasis I try to say in all of my lectures, and I've been doing this now for decades more in teaching biochemistry. All right. Now, more detail. Okay. There is a molecular elasticity and plasticity, and we see this in the stability of T regulatory cell lineages. And that occurs via transcription factor binding to what are known as conserved non-coding sequences. Unfortunately, those are called CNSs. You can see down here in the chromatin. You have CNS naught, CNS1, CNS2, and CNS3. In their association in the gene that's going to code for FOXP3, ultimately. That global transcription factor for just one of those T cells I told you about, which suppresses the immune response, at least suppresses the inflammatory immune response. And those are the T regulatory cells expressing high levels of FOXP3. And also all those other molecular signatures I mentioned, and many more now you're going to see. Okay, so now we're getting more to the detail of just T regulatory cells, because these T regulatory cells and their function are what often fail during disease. There's a lack of T regulatory cells, so you have runaway inflammation, which is what I was talking about with chronic diseases associated with obesity, for example, and linkages to those obesogenic states, and even the non-obesogenic states that can lead to things like atherosclerosis, right? Overall cardiovascular conditions, or those associated with oncogenesis, the uncontrolled proliferation of cells leading possibly to tumors. Right? So you want to have lower levels of Treg activity in a tumor because you want the tumor destroyed by the inflammatory T lymphocyte lineages, don't you? Yes, you do. Whereas you want Treg activity when you have, for example, an inflammation because, say, you had a TIA in the brain, right? And that ischemic activity in the brain led to an inflammatory response, and you want the T regulatory system to shut that down. Well, in the brain, you're very limited in the amount of T cells that are available. So the microglia, which are functioning as a border between the innate and the acquired immune response, those are the major immune cells in the central nervous system. We'll talk about those later, but I will remind you, I've talked about them in great deal over the last year. So you can go back and listen probably to 10, 15 different lectures just when I was talking about microglia, right? So that's what I warned you about. When we get to the point of doing these synoptic video lectures, I'm going to expect that you know a lot more than what I'm presenting here today. And I know that you don't necessarily have it all running around in your prefrontal cortex or ready to write it out for me in a 15-page answer to a question such as, what are 3 possible ways that T regulatory cells can malfunction over time as with human aging? Okay, I would expect 15 written pages on that, and even with an open book. Right? Of course, open book would be a take-home exam. Okay, so let's go through this. Molecular elasticity and plasticity to stabilize Treg via transcription factor binding to conserve non-coding sequences for chromatin retailoring. See, I dropped the chromatin remodeling. I'm not going to use it anymore. Chromatin retailoring, which is necessary simply to procure FOXP3 expression. That's all this is. This whole thing I'm talking about now. So you have epigenetic modification of cohering DNA and histones in the nascent CD4 positive T lymphocytes. And that will conduct a differentiation to the Treg lineage because you're going to be making FOXP3, which is the cognate molecular transcription factor signature for T regulatory cells. And then all that downstream immunology and physiology, you see. Now, all of this is coming from a paper. I didn't mention the last ones that we were looking at. This is Frontiers in Immunology, but it's already 6 years old. Now, check out what's happening here. First of all, you have to engage the T cell receptor, which is itself a product of a recombination of multiple gene fragments, just like we get with immunoglobulins in B cells. And remember, we talked about the education of T cells in the thymus and how any T cells that auto-react or auto-self-react because of their TCR recombinant, they will be eliminated, right, by negative selection, we call that. And I'll go into that in more detail here again, because we don't want any T cells that are going to react with self-antigens. Right? So let's just say that these are CD4 positive T cells that have a TCR that's already been to undergraduate college and has learned the TCR it has will not or should not respond to host antigen. So it's not going to become self-inflammatory, right? Or self-regulated, which we don't want. But T regulatory cells have TCRs, and with associated with them is another protein called CD28. All right, so look what happens here. You're gonna need all of these processes here and all of this epigenetic alteration just to make FOXP3, the transcription factor, which is the signature for Tregs. So you got here TCR receptors engaged. We won't go into that whole process. I removed it entirely, and plus this figure from this paper didn't have it either. But you induce a TCR response and associated with CD28. You turn on phospholipase C gamma, which is going to release calcium stores. That's going to induce calcineurin, and at the same time, it's going to cause the removal of phosphate from a protein called N-fat. And those phosphates are going to be removed. So now once that phosphorylated protein called N-fat is dephosphorylated, covalent modification change from kinase to phosphatase, because of the TCR recognitions, for example, of antigen presenting cell. Said I wasn't explaining what's going on there, but I had to. And then PLC gamma, releasing the calcium stores, this phospholipid metabolism right there at the beginning, as phospholipase C gamma, Okay. Phospholipase C is going to generate diacylglycerol. Right? You're going to have the calcium-calcineurin. You're going to cause this phosphorylated protein and fat to lose it, and fat will then be able to enter the nucleus. That's important because you need N-fat as a component of the transcription factors. Okay, it's a component of the transcription factors. All right. Now, along with that, phospholipase C gamma is going to remove, for the similar, because of a phosphatase activation, it's going to allow P38, ERK, and J and K, those phosphorylated proteins, to generate AP1 in the nucleus, to mobilize AP1 in the nucleus. So now you have N-fat, you have C-rel, which is already in the nucleus. You have protein kinase A induced by the phospholipase C gamma. Diacylglycerol did that. Remember, DAG to pKa. That's going to associate CREB, because it's going to be a cyclic AMP response element binding protein, to ATP. And so now you're going to have an SP1 protein, a dimer of CREB, in the presence of a protein called RUNX and CBF beta. You're also going to directly then generate transmission of the FOXO13. That's not FOXP3. What else is happening? Oh, it looks like I'm getting dark here. My lighting engineer isn't in. I'll have to change that in a second. In the presence of interleukin 2, we were just talking about, which was being synthesized, remember that, from the early stages of the plasma cell, for example, binding to the receptor, turning on the STAT pathway. So you dimerize this protein STAT, and it moves into the nucleus. And one more thing happens when the receptor, interleukin 2 binds its receptor. The SMART protein will block STAT3, because STAT3 would shut everything off and prevent this dimer from mobilizing into the nucleus. So now you have STAT, dimer, phosphorylated, FOXO13, RUNX, the CREB, SP1, N-fat, and then also induced through here is AP1. And then one more thing to mention, the TGF-beta receptor, because remember TGF-beta is also generated during this inflammatory response. These are all Treg cell mediated events, as I've been saying. It's going to work through two proteins called SMAD2 and 3, and you're going to phosphorylate SMAD2 and 3, so then it can react and make a trimer. So three proteins functioning together, SMADs 2, 3, and 4. That will allow all the SMADs to move into the nucleus phosphorylated. That's what that means. At the same time, TGF receptor is going to get RUNX1,3, the TEAG1, also all in the nucleus. All these proteins now are made up. It's like they've assembled themselves in the nucleus based on this triggering phenomena. So now we can look at the promoter for FOXP3 finally. The FOXP3 promoter has 3 conserved regulatory regions, and the epigenetic modification status of the FOXP3 gene is going to be shown ultimately along these peripheral changes. So upon TCR stimulation, just repeating what I just said, N-fat, AP1, SP1, CREB, ATF, it wasn't ATP, there was ATF, bind to the promoter of the FOXP3 gene. Okay. So all of these proteins are going to be binding to the promoter. There's the FOXO13, there's the SP1, there's the dimerized STAT, there's N-fat, there's C-rel, AP1, there's the RUNX1,3. All on the promoter. Remember, that's going to be 5' proximal for the open reading frame for the production of FOXP3, the actual gene transcript product. This is all in the promoter region. This is just on the chromatin minus 2b minus 2a, or 2 significant reasons. Because right after 2a, this is again before the open reading frame, you have these 3 CNSs, right? Conserved non-coding sequences, 1, 2, and 3. And they have also more ornamentation of all of these cohering transcription factors, just to make FOXP3. There's more N-fat, SMADs, the phosphorylated SMADs. Remember, we talked about those. AP1, TEAG, and then in close association is CNS2, non-coding sequences now. The CREB proteins bound, the phosphorylated STAT, like on the promoter, is bound here. Several of these other co-adapting transcription factor um, cohering proteins, right? All come together right about when you get to the transcription start site, but you're still not there really because you have one more CNS, non-coding region, and that's got to bind CREL. Okay. So STAT5 forms a dimer, response to interleukin-2, we talked about that. In the periphery, TGF-beta signals drive the SMADs and the end-fed occupancy of the CNS1 position and can induce FOXP3 expression. It's in the periphery, okay? So you have thymic FOXP3 expressing Tregs, and you have peripheral. We talked about this a couple of lectures ago in the audio, okay? Now you have a CPG island within the FOXP3 promoter region, and it's going to be demethylated, okay, in both thymic Tregs and in the peripheral Treg cells, but not in naive T cells. This gives you now this molecular signature, demethylation. Okay, so when you have a methylated CPG island for FOXP3, you don't get expression. So the Treg doesn't differentiate. So you don't get the suppression of the inflammation. You see? Okay. The histones bound to the FOXP3 promoter, okay, in this region here. Remember, these are basic proteins involved in the chromatin. Bound to the FOXP3 are hyperacetylated at this point, and you find that hyperacetylation, the substrate for that is acetyl-CoA, and the enzyme is the histone acetyltransferase, so the HAT proteins. And you get that both in TT-regs and PT-regs for the induction of the full Treg lineage either in the thymus or in the periphery. CNS2 also has to be demethylated, but you only see that in the thymic Tregs. It's not necessary to be involved in the peripheral Tregs. So it's just showing you more detail. The naive CD4 T cell has a methylated CPG island in that promoter, and both now forming Treg populations, the thymic and the peripheral. You have demethylated CPGs and acetylated histones. The CNS2 you had methylated to begin with. That's a naive T cell, but now it's differentiating into Tregs. Again, demethylated CPG, acetylated histones. And in the PTreg peripheral, you still have a methylated CPG there. It's a slight change in regulation between what's happening in the thymus and what's happening in the periphery. Okay, give me 1 second. I'm going to have my photic engineer turn the light on. Hopefully that's better. Yes, you can see me again. All right, let's continue. Thank you, lighting engineer. All right, so where are we at right now? Here's a paper that was published in 2020, The Frontiers in Immunology. Okay. Now, the papers we've been looking at so far today are open access. Many of the other papers that I like to use are not open access, but some of these, and I'll put these in the show notes eventually, you can read the whole paper because these journals give open access. But you'll find out, and I'm sure all of you know this that are scientists, and maybe some of you MDs, that probably half of the papers that you really need to get at are behind a paywall. And so you need to have some kind of subscription to a library that has those journals, right? And then the other half of all published scientific research is open access, which means anybody can get it, just using PubMed to find out what those papers are. So I'm just explaining to you a little bit about the etiology of getting the papers I'm examining. Right? I think all of you can understand what that means. So this paper, Frontiers in Immunology, Treg transcription factor FOXP3, genetic mutations. Okay, so we went through that whole Treg so I can get to this point about the dialectical event ontology of T lymphocytes related to the aging process and some frank diseases. That's where we got this far, so far in this lecture. So Treg transcription factor FOXP3 genetic mutations lead to autoimmune disease. Now you can understand why. Simple reason. if Tregs suppress inflammation, and autoimmune diseases have to do with uncontrolled, not always, but often uncontrolled inflammatory responses. If you don't have Treg activity, you're going to have a chronic inflammatory response. See? Now you understand that. Now, FOXP3 positive CD127, negative CD25 high, those are three different proteins, obviously. That's the full molecular signature for a Treg. FOXP3 transcription requires the functional and persistent T cell receptor stimulation. I just showed you that via an APC, presumably. You can also get TCR stimulation, of course, with complement associated with B cells and with immunoglobulin. And we talked about that also recently. So the analysis of FOXP3 positive versus FOXP3 negative T cells, according to this paper published just last year, suggests that many Treg specific genes don't actually require FOXP3. So I went through this whole thing with FOXP3 being an absolute molecular signature for Treg, and that's true. But there are a lot of other functions for Treg cells. I told you they were immunomodulatory, not just suppressive. That's why they're called Tregs, right? Regulate doesn't give you the valence. It just tells you they regulate. All of that suggests that FOXP3 is an either-or protein in Treg signature activity. Now, by that, I mean I take either-or from actually a book written by Soren Kierkegaard, where he talks about the three possible views of how to live through life as an existentialist. And I'm not going to tell you what those are because I don't want to get dark into that detail. But either/or doesn't mean only two choices, does it? Either, let's say, polar, nonpolar, or, see, either or hydrophobic. Right? So you have three choices. You have an either choice, which is a distinction, a dialectical distinction between two things, or a third. Right? And that's what I'm telling you we're going to discover with FOXP3. Now, why is all this very stark molecular discussion important for you to understand aging? It's because this is what you must understand to see what occurs in the aging human. What kinds of things at the molecular level start to fray and disintegrate in terms of their normal, healthy regulation. Well, there are many, many, many things. Like I said, if you look at all the interactomes, right, and all the individual events, you're looking at hundreds of millions of different interactions, right? But we can't talk about all those at once because we can't look at the entire dynamic movie that's happening all the time and say, this is where the change is happening here in this one small subset of subsets. So we try to move up the hierarchy and look maybe if we can get this good at cell lineages. And that's why we look at Tregs, because we know that Tregs can either stop, for example, an inflammatory response or let one go, or they can modulate. And the modulation is not off on, it's something else. Hence, FOXP3 is not the only transcription factor. You understand? All right. So I'm going to stop here because now I'm going to start getting into, as you can see, I'll move myself here, more frank discussion of this FOXP3 locus, how all these different proteins interact, and then I'm going to get into the epigenetic control in more detail. You can see we're already going to be talking about histone acetyltransferases, and we're going to be talking about HDACs, those are deacetylases. These are all the enzymes. These are all of the mechanics for the epigenome. And again, these are DNA methyltransferases. So all of the, just looking at acetylation, methylation is part of the epigenetic signature we're now going to superimpose on all that regulation we just talked about with all those different protein cofactors to just induce FOXP3 expression, which will give you the professional Treg cells, with the caveat and understanding that Treg cells working in the either category, on or off in terms of suppression, there's an OR associated with that, and that OR is immunomodulation, which maybe has very little or nothing to do with FOXP3. So thus opening up the diversity of interactions that must be understood to see how this links to what happens in the aging human, particularly the modulation, alteration of T regulatory cells. And then we'll talk about the other T cells and move up more and more. So I'm going to stop here because I think I probably spent enough time today examining what I think is a really important feature of this whole system. There we go. And what I mean by that, to close, is what I'm trying to do is give you the immunological axis in that trigonal planar descriptor of the aging human as a paradigm for an understanding of how looking at a dialectical interpretation of two competing, either contrarian or maybe even appear to be pseudo-contradictory modes of interaction have to be embedded within our understanding of those interactions with the genome, the environment, and the immunological axis. And that immunological axis is heavily defined by epigenetic alterations. Now that ultimately leads us into the aging process, what we observe in the aging human, the loss of hair, the hair turning gray, hair falling out, right? Loss, I guess I've already said that, but also the whole process of the physiology, the normal physiology, starting to degrade into pathophysiology in various regions, both in the periphery and the central nervous system, ultimately leading to a series of morbid conditions that can lead to death, right? So that is now how I'm going to end this. So let's get back to where we were. And I think I can stop it here.